Welcome back, students. Today we are going to discuss part 5 of Unit 2, that is Unit of Life. But before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator, owner of these images. If you are the owner of any of the images used in this particular PPT or have any concern, kindly contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss some points related to the cell, like what is cell, what is the cell theory, and overview of the cell membrane, and active and passive transport system. Okay, so as you know, cell is a basic unit of life, and all the living organisms present on this earth are, this is the basic and fundamental unit. Okay, they are all composed of cells, right? Some organisms are, composed of a single cell. Only single cell are there. That time we can use the term unicellular for them. Okay, unicellular. While other are composed of many type of cells, like multicellular organisms, like human, plants, animals, etc. Okay, and in unicellular, what kind of type of organism? Bacteria, amoeba, right? They are under this particular category. Okay, so unicellular organisms are capable of independent existence and performing essential functions of life within single cell. Okay, so anything less than a complete structure of cell does not ensure independent life. Okay, so can we say this, that cell is a fundamental structure and functional unit? Yes, we can say this, that cell is the fundamental structural and functional unit of all living organisms. Or in other words, we can also say that cell make up living things and carry out activity that keep a living thing alive. Am I right? Okay. So let's discuss some points related to the discovery of cell. Was the first person Anthony van Leeuwenhoek. He was first saw and described a live cell. Okay, and Robert Brown later discovered nucleus. Hmm? And how it happened? With the help of microscope only. Okay, so invention of microscope and its improvement leading to the electron microscope reveal all structural detail of the cell. So this microscope helped us a lot. Anthony van Leeuwenhoek saw that first cell under light microscope only, right? So actually cell are too small, micron size. Okay, micron means 10 to the power minus 3 millimeter. Okay, so cells are too small to be seen by naked eye. So they are studied with the help of microscope only. Microscope are high resolution instruments that are used for observing fine details of very, very small objects. You must use microscopes during your 10th and 12th class journey, right? So this is the use of microscope. Now let's compare two type of microscopes which are present. One is light microscope, another one is electron microscope. Light microscope, you can see this type of microscope in the school colleges as well, while electron microscopes are normally present in research laboratory. Okay, so electron microscope have high resolution about 0.1 nanometer, 0.1 nanometer, which compared to about 200 nanometer for light microscope. Can you see the difference? So with the help of microscope, electron microscope, we can also analyze any part or anything present inside a cell in nanometer level. Okay, now let's discuss some points related to the shape and size of the cell. Shape and size of the cell vary but all of these are ultimately determined by their specific function. Like some cell can change their shape. Yes. Is it possible? Yes, it can be. Like amoeba is one of the very good example. And amoeba can change its shape. WBCs can change its shape. But if we are talking about plant and animal, they are not able to change their shape. They have fixed type of structure. Right? So, but some cells can change their shape. This mycoplasma, or popularly known as PPLO, this is comes under this category, smallest cell. So if any question related to the smallest cell, you quickly can answer PPLO, right? And what type of cell comes under largest category? So egg, ostrich, sorry, ostrich egg. That is a single cell and largest cell. Okay, and the longest one is the nerve cell. These few example are given in your NCERT book. That's why I have also quoted this over here. Okay, see, there are different shape of different kind of cells. This is nerve cell. This is your mesophyll cells, round and oval shape. This biconcave cell, biconcave structure of RBC, or we can say erythrocyte, platelets like this, white blood cells, almost circular, basophil, neutrophil, eosinophil, monocytes. So different type of cells are there according to their function, right? Okay. So let's compare unicellular organism and multicellular. This unicellular are composed of single cell only. This term define itself, right? While multicellular composed of multiple cells. So in unicellular, single, or we can say if only one cell is there, then body must be simple. So simple body organizer. And this one is complex body, right? Total cell body exposed to the environment of, because only one cell is there. So total cell of the body exposed to the environment, while in the case of multicellular, only cells present on the outside surface. Like in human being, only skin cells are exposed to the outer environment, right? What are the examples? Amoeba is one of the example. Paramecium is another example. These all type cells comes under this particular category, while multicellular plant cell, animal cell comes under multicellular category. This simple or unicellular cells cannot divide into further cell because they contain only one cell. They are also called, that's why they cannot divide into the cell. That's why we can use the term acellular organism for them. This is an important point. Acellular organism. Clear? While in this case, tissue, different kind of tissue form organ, different organ come together and form an organ system, right? To perform a specific type of function. Okay. So in broadly, these are the main difference between unicellular and multicellular organism. Now, cell theory. Okay. So there were two scientists, Leiden and Swan. One is botanist and another one is zoologist. Together formulate the cell theory. Okay. There were another very renowned scientist, Rodolphe Virtue. In 1855, he was the first person to explain that cell divides and new cells are formed from pre-existing one. And this is a very famous quote he used, omnis cellula e celluli, means new cells are formed from the pre-existing cells only. Okay, so he modify this virtue, modify the hypothesis of Slider and so on, and give a final theory of this cell theory, which is popularly known as cell theory. That first point is like this: all living organisms are composed of cells and produce and the product of cell. All the living organisms. Okay. And second, all cells arises from pre-existing cells. First point, all living organisms are composed of cells and products of cell. Second, all cells arises from pre-existing cells. Okay, so this is all about the cell theory. Now, different type of cells, prokaryotic type of cells, bacteria comes under this particular category. Eukaryotic type of cell, any multicellular, any plant cell, we can say animal cell comes under this particular category. There are multicellular organisms. Okay, the what is the outer covering? Cell membrane, and in the case of plant, cell wall is also there. Different kind of cell organelles are there. We will be discussing in the coming video lectures, like chloroplast, mitochondria, vacuole, endoplasmic reticulum, Golgi complex, nucleus, what kind of structure they have, what is the function they are mainly involved in. So we will be discussing it in the coming video lecture. But in today's video lecture, we are going to discuss about cell membrane. Okay, but before that, we must understand if this is a cell. So the outer covering is known as cell membrane, and there is a fluid inside. This whole structure is filled with a fluid structure that is known as cytoplasm. Cytoplasm is present inside it, and there is a comparatively condensed area known as nucleus, and this nucleus is mainly responsible to control all the cellular activity, right? And plasma membrane actually create a separate environment and give cell a type of structure, and mainly responsible for the transportation purpose only. And the cytoplasm is present inside the cell. Let's see this one. This is cytoplasm. Okay, this all kind of metabolic activity occur over here. Okay, so it is very general type of diagram I have drawn just for you to understand all the main content of the cell. Just outside, plasma membrane, which actually separate internal environment with the external environment. Okay, nucleus is for its control, its activity control, and cytoplasm possess different kind of metabolic machinery. Okay, now let's discuss biomembrane. Okay, so there were different kind of models has been proposed by various scientists. Okay, so let's discuss this part first. First model is was proposed by scientists is this lipid bilayer model. Lipid bilayer model, and according to this model, this any type of plasma membrane is composed of lipid bilayer. Say for example, this is a lipid layer and this one is another lipid layer. Okay, so according to this proposal, lipid bilayer model, any type of plasma membrane is composed of lipid bilayer. But this one is not accepted by the scientific community. Next model came that time, and then that was known as Davison and Daniel. These are the two scientists who actually proposed this model, and according to their model, two layers of protein flank the central phospholipid layer. Means there is two layer of protein, external side as well as internal side, like this. This is protein, and okay, this is lipid bilayer according to Davison's model. This model suggests that on both the terminal, external and internal, protein is there, and these protein flanked a central phospholipid phospho. Oh, a second. Phospholipid layer. Okay, according to this. So, lipid bilayer suggested that all this plasma membrane is composed of lipid bilayer. Davison suggested that no, not only lipid, protein is also there according to him, and this phospholipid layer uh, was flanked by the protein, and we can also use the term sandwich model for the same. Sandwich model. After that, Robertson model came. Okay, or we can use the term unit membrane model for the same. Okay, and according to Robertson model, biological membrane is a lipid bilayer surrounded by the protein, just like Davison model, from external and internal side, both the side. Okay, and the one important point he added that time, that the both these type of protein are hydrophilic. Okay, so according to him, hydrophilic content are present on the outside, and hydrophobic content means lipid bilayer present inside of this content. Means same type of model. I can means if I'll write over here hydrophilic, and this lipid, of course, hydrophobic. So like this, he just changed the structure somehow, but this his model was influenced by the Davison's model. Okay, but this was also not 100% accepted by the scientists of that time. And the finally most accepted model is known as fluid mosaic model, and this was given by Singer and Nicholson in 1970. We will be discussing about this model in detail. Okay, clear? Okay, now, so fluid mosaic model, the outermost layer, which one is elastic? Okay, it cover the cell and separate the cell content from the external environment, is called plasma membrane. Okay, the outermost layer of the cell is a membranous covering of the cell, and it separates cell's content from the external environment, and this is known as plasma membrane. And this plasma membrane is selectively permeable. This one is selectively permeable, means it allow entry of certain substance only, restricted entry, and it is made up of lipid bilayer and protein, and proteins are integrated in out and inside out. Some small amount of carbohydrates are also attached to these proteins on the outer surface of lipid, not protein, on the outer surface of the lipid and protein both. Okay, so let's see the structure. This is the structure of fluid mosaic model. Okay, so this is the lipid bilayer. Lipid bilayer, hydrophilic head towards on the top side and hydrophobic tail on the middle side. And this lipid bilayer act as a C, and in this particular C, some embedded proteins are there, embedded. Just like eyes, this is integral protein. Okay, some peripheral proteins are also there. Here, this is the peripheral protein. This is the peripheral protein you can see over here. And some carbohydrate chains are also attached to the lipid and carbohydrate. Lipid and protein. Right? So this is the most accepted model given by Singer and Nichols, known as fluid music model. Why it is known as fluid music model? Because it looks like a fluid sea in which ice works, just like a protein act as a ice and it float in the a sea of lipid. That's why they gave the name fluid music model to his model. Okay, so these protein which are there, peripheral and integrated one, can act as a receptor, they can act as a channel, they can utilize for pump purpose, anchor purpose, or sometimes they can also used as a enzyme. Some biomembranes also have carbohydrates, as you can see just now in the picture, attached to the lipid or protein, and they are forming glycolipids or glycoprotein. That time we can use the term glycolipid or glycoprotein. These carbohydrates actually, what is the role of these carbohydrates? They actually serve as a recognition marker for other cell or molecule or as a protective coating. Okay, so this is again a very important function performed by the carbohydrate molecule, right? The biomolecules are not uniform in their composition and function in all type of cells. They can be different according to the organ type, okay, according to the cell type. Means the ratio of lipid and protein and carbohydrate may vary according to the type of organelle, type of cell. Okay, yes. So let's discuss some functions performed by the plasma membrane. They act as a selective barrier, okay, allow only certain substance to pass through, mediating communication and signaling between cells or within cells. Second very important function performed by the plasma membrane. Third one, hosting specific enzyme and transporters that catalyze chemical reaction or move molecule across the membrane. So act as a transporter as well. Maintaining the shape and structure of the cell or organ. Okay, so all four functions are very important. These are performed by the plasma membrane. So as just now we have discussed, this plasma membrane also act as a transporter, so means it involved in the transport system, right? So one of the most important function of the plasma membrane is transport. As you can see, there are two type of transport. One is passive transport and another one is active transport. Okay, passive transport, when certain kind of molecule come from the high concentration to low concentration or along the concentration gradient. Okay, so diffusion is one of the example, and if they pass from one outer external environment to the internal environment, they then at that time they must use a protein or transporter. Okay, so these two are the examples of facilitated diffusion when they take out the help. Active transport, when energy is required for transporting any molecule. So of course, if energy is required means it occur against the concentration of gradient, right? So let's discuss all three types in a little bit detail. First is passive transport. Just look at the definition. Passive transport does not require energy. Just now you have seen in the picture, it doesn't require energy input from the cell. It occurs naturally. Why? Because of the concentration gradient, right? First type, simple diffusion, where small molecules, non-polar molecules move freely across the cell from the concentration of high concentration area to low concentration, right? Second type is facilitated diffusion, where large molecule or polar molecule, okay, large molecule or polar molecule use protein channel for its own transportation. They must use some kind of protein channel, some kind of carrier molecule to transport themselves from outside to inside or along their concentration gradient. Okay, that's why we can use the term facilitated. They need a help. Okay, the third type is osmosis, right? Osmosis Also, it is a spontaneous movement of solvent molecule. Okay, solvent molecule through a selectively permeable membrane from a region of high potential gradient to low water potential, or we can say in the direction that tend to equalize the solute concentration on two sides. As you can see over here, this is a semi-permeable membrane. Okay, concentration of solvent is more over in here. In dark blue dots will move towards the that area where concentration of solvent is less. Okay, then after some after certain time, what will happen? Mostly solvent molecule will pass the membrane and come towards this side. This one is 1 and 2, 1 and 2, first jar, first part or second part. So in this situation, what will happen? The total volume has been increased, okay, of this size, right? This volume has been lowering down. Why? Because mostly solvent molecule cross the semi-permeable membrane and come on this side where solvent concentration is high and solvent concentration is less. Okay, so this is again a spontaneous process and net movement of, or we can say diffusion of solvent molecule occur. Okay, now let's discuss active transport. Okay, so active means energy utilization, or in a process in which a kind of process where ATP is used. Okay, so active transport require energy input, usually in the form of ATP, to move substance against their concentration gradient. Why we need energy? Because that time we need to move against the concentration gradient, means from low concentration to high concentration. As you can see in this particular picture, the concentration of this substance is less in here, in outside cell, and inside cell this uh, the concentration is in ample amount, but this particular molecule need to be come inside the cell, so against concentration gradient. So in this situation we need to use ATP. That's why this type of transportation is known as active transport. Okay, we can further divide active transport into two different categories. First is primary active transport, second one is secondary active transport. Primary active transport, in this process energy is directly used to pump molecule across the membrane using proton pump, like a very famous example, sodium potassium pump. This example comes under primary active transport category. Secondary one, this type of transport couple the movement of one molecule against its concentration to the movement of another molecule down its concentration. Okay, and what is the example? Sodium glucose transport. Sodium glucose transport is the example of secondary active transport. Clear? Okay, so this is a picture showing sodium potassium pump. Concentration of sodium is high outside the cell. Okay, this one is plus outside the cell, and potassium, concentration of potassium is present high, in a high concentration at inside the cell. So high to low. Okay, high to low. So this one is required, not high to low, but low to high. I'm so sorry, low to high. See, so low to high, then we require ATP for this kind of reaction, right? In secondary pump, what will happen? If sodium is more present in the outside, so automatically it come towards the cell. Okay, and that time what will happen? Energy is there, but with along with sodium come glucose is also transported in either way, whether the same direction or in opposite direction. Okay, we will discuss that topic later on in the next video lecture. Right now, I am concluding this particular lecture in which we are discussing about cell theory, different type of cell, difference between multicellular, unicellular, and different type of plasma. I hope you understand all the topic. Still you have any query, any doubt, you can write down to me comment box. We will discuss later. Okay, till then, take care.